Hello and welcome to another Pelago webinar. I believe we can just wait a couple of minutes. There's still some people joining. In the meantime, my name is Renato Alves. I'm a project advisor here at Pelago Bioscience. And I'd like to thank everyone for joining us in this webinar today, which is titled Fungal Genomes in Drug Discovery, Application of CETSA to the Study of the Mechanism of Action of Brefeldin A. Before starting with the scientific part, the presentation, I would like just to do some housekeeping. This webinar is being recorded and it will be uploaded onto our website within a few weeks. The presentation of the scientific part will take around 30 to 40 minutes, and after that we will have a Q&A session for which you can use the chat function to ask your questions. You're welcome to drop the questions along the presentation, and then we will address them in the end. Today's speaker is Ben Fontaine. Ben got his PhD from Emory University in the US, and he later did a postdoc at UC Berkeley in the lab of Professor Dan Nomura, where he used chemoproteomic technologies to develop novel modalities in targeted protein degradation. Currently, Ben works as a senior scientist at Lifeline Therapeutics, where he applies chemoproteomic profiling in conjunction with CETSA to deconvolute target profiles of fungal natural products, opening the way to novel nature-derived medicines. Before we start with Ben's presentation, I would just like to introduce a little bit more about Pelago. So our passion here at Pelago is target engagement, and we are passionate for showing target engagement in a physiologically relevant condition. So CETSA as a technology can be applied throughout the whole drug discovery process, and it provides you with relevant quantification of compound binding to a protein of interest in the intended site of action inside the cell. So we are connecting chemistry and biology at the relevant site of action. CETSA can also be applied in a variety of steps, namely to generate tool compounds or active compounds in studies for selectivity and specificity, biomarkers and translational studies, as well as for the initial target identification or validation. And we provide you with biologically relevant data that will enable faster and more precise decision-making in your drug discovery process. We are basically linking the initial steps. So as soon as the drug and the target become in close contact with the later phenotypical responses that many times is the starting point for your drug discovery program. And Pelago can help you with basically understanding the whole timeline that goes from that initial drug target binding in the initial first seconds until the later transcriptional response, new protein synthesis, etc. CETSA, which is what brought us here and what made Pelago what it is today, is a thermal shift assay done in live cells. And it's based on the principle that when we increase the protein, when we increase the temperature, the proteins will start to unfold and denature and precipitate. But if the proteins are actually bound to a drug, they may require a higher temperature to start to unfold and denature. And it's that difference between the naked protein that hasn't been treated and the protein that has been treated with the drug and it's bound to that drug that we are measuring, and that's the shift that we are measuring in the thermal shift assay. Of course, it can also happen the opposite, where by actually being bound to a drug, instead of requiring a higher temperature, the protein actually requires a lower temperature to start melting and precipitating. And that is equally a sign of target engagement. And it is also measured by CETSA. The greatest advantage of CETSA is that it can be applied to a wide matrix, a wide range of matrices. It can be applied to human, animal, plant, fungi, as we have done before in the past, and today's presentation slightly touches that area. But it can also be applied to cell lines, to primary cells, to solid tissues, and we can use it either in intact cells or on a lysate or homogenate. Our portfolio includes 4 different formats of CETSA, which can be applied into different stages of your drug discovery process. 3 of them are actually targeted CETSA, where we are actually looking at just one target, and then one of them is a proteome-wide CETSA analysis, looking at using mass spectrometry and looking at the whole proteome, and that is actually the format that today Ben is going to present to us. Apart from CETSA, Pelago can offer a wide variety of other assays, namely plate-based assays for screening, toxicity studies, mode of action, etc. We pride ourselves to design our studies tailored to the client's needs, which require that we have a initial conversation with the client to understand what are their needs, how can we help them, and then in the end, a new project which will be adaptive to each client and their needs will be conducted, hopefully to get the client to the place where they would like to be. Pelago works mostly as a fee-for-service, although we also are working more and more on an FTE-based model. Because we control the intellectual property on CETSA, we can also offer licensing agreements that can go from licensing the whole CETSA platform to a single target license on just one assay addressing one single target. And of course, we always have strategic partnerships that we are eager to initiate when we see that there's a shared value creation between our partner and Pelago. Pelago is also located in Sweden, in Stockholm. We have recently, around a year and a half ago, moved into new facilities, 3 times the size of our previous facilities, and we now have about 2,000 square meters divided into half of lab space and half of office space that we can use to deliver the excellence in our projects as we like to do. But without further ado, I think we can pass to Ben and to hear his presentation. So I'm just going to stop sharing my presentation and allow Ben to start sharing his.
All right. Thanks a lot, Renato, for that great introduction about my career and also a very nice introduction into the CETSA technology. Let's see if we can get the share going here. Okay, I think we should be good to go.
Yes, perfect.
Cool. Great. So let me pull up the pointer here. And yeah, so thanks again to Renato. I also just want to quickly thank Stina Lundgren as well, who gave me the opportunity to come here and present at this webinar. And thanks to Tomas and Alexi for really helping to design the study, and also to Tomas for getting into the lab and doing all the heavy lifting to generate some really nice data on this fungal natural product, Brefeldin A, that I'm really excited to tell you guys about today. And finally, thank you to Jacob and Bora for helping to organize this event as well. So I will start off with a little bit about who we are and what we do here at Lifeline. We are a preclinical private biotechnology company located in beautiful Cambridge, Massachusetts. And our secret sauce here is that we are developing and deploying proprietary algorithms to mine the fungal biosphere for precision medicines in the form of natural products, or as we call them, genetically encoded small molecules, or GEMs. The company was founded a little over 5 or so years ago by Greg Verdine, who's a professor emeritus at Harvard. He's founded several other biotech companies over the years, along with Wei-Ching Zhu and also Rick Klausner, who was the former head of the National Cancer Institute here in the US. And we're spread out across 3 sites. The bulk of our R&D activities take place right here in Cambridge. We also have a smaller site in Gloucester that handles a lot of the routine maintenance for our massive fungal strain collection. And we recently launched a site in Basel, Switzerland, right here along the beautiful Rhine River. And there we've got a good mix of everything from synthetic chemistry to discovery biology and also synthetic biology, as well as some other activities. And we're sitting right at around 100 or so plus employees at this point. So our platform for drug discovery is called Avatar RX, and I'll explain in the coming slides what exactly that means and what exactly that name stands for. But for now, I just want to give you guys a very high-level overview of our very unique approach to drug discovery here at Lifeline. It all starts with what we call LifeBank, which is our massive strain collection, which is now over 100,000 unique fungal organisms. And we've acquired full genome sequences on all 100,000 of these strains, which we've assembled into a digitized searchable gene pool called Lifebase. And then we're constantly developing and improving intelligent algorithms to search these biosynthetic gene clusters for tailor-made genetically encoded small molecules that we can then think of as advanced leads and further convert into drugs against human diseases. And of course, the isolation here in this pipeline to actually pull out these molecules relies on state-of-the-art genomics, metabolomics, and transcriptomics to identify the candidate compound. And then we can go in and confirm on-target activity using target-centric assays. And we have a huge amount of complex molecule synthetic chemistry expertise to explore the SAR space on these really challenging molecules as we seek to convert them into clinical candidates. So a little bit about our strain collection LifeBank here. This has been assembled through the acquisition or the licensing of several different collections, as you can see here, including the large strain collections from both Pfizer and Merck. We've also recently acquired this nice Australis collection. We're also supplementing the strain collection internally through our own collection efforts. A lot of this goes on at our site in Gloucester, and we actually have a nice little sort of fungal collection kit so that, you know, our employees can go out and try to mine the soil for different fungi, and then we can try to clean those up and put those right into the collection and into the genomic database as well. Okay, so one question that we get all the time is, why are we exclusively focused on fungi? Why don't we dabble in plants or in bacteria or anything else? Well, the reason is, as humans, we are remarkably similar to fungi in terms of our the conservation of our protein sequences. So we're much more closely related to fungi than fungi are to plants. I think that that fact gets lost a lot of the times. So what this means from a practical perspective is that genetically encoded small molecules or drugs derived from fungi often target the very same protein across both these kingdoms of life. A classic example of this is the cholesterol-lowering agent lovastatin, which inhibits HMG-CoA reductase. So, of course, the fungi aren't making this to help each other lower their cholesterol. They're doing this because it hits a critical node in sterile biosynthesis, and it acts as a potent antifungal agent. It turns out, as humans, we can take this very same molecule, it inhibits the exact same target in us, and we get a very nice cholesterol lowering effect. So a blockbuster drug here. Another hallmark example is the immunosuppressant cyclosporine shown here. This molecule inhibits the phosphatase activity of calcineurin in both humans and fungi by forming a molecular glue complex with this small protein called cyclophilin. So maybe some of you can see where I'm going with this. If we have methods to intelligently predict the fungal target of a given gem, we can then effectively predict what that molecule is going to target in a human setting as well. So before I dive deeper into our platform at Lifeline, I just want to take a second to remind everyone of how natural product or gem discovery was done in previous years. It really relies on this brothology or activity-guided fractionation approach, which starts with some typically ungodly complex extract containing hundreds to thousands of different natural products. And then these are then iteratively fractionated using some sort of functional assay. So many times this will be a phenotypic assay where inherently we have no knowledge of the mechanistic target. But in any case, these assays can be used to guide the fractionation until we ultimately isolate a purified gem responsible for the bulk of that desired bioactivity. And with the advancements in genomics and transcriptomics and metabolomics, it really doesn't make sense to be doing natural product drug discovery in this way any longer. So that's why we've pioneered our platform here, Avatar RX, which enables a target-centric approach to natural product discovery. And we can leverage this to pursue targeted search and retrieval campaigns for genetically encoded small molecules that have evolved specifically to engage a given human therapeutic target. Effectively, what we've done is we've extended the central dogma of molecular biology to deep mine fungal genomes for genetically encoded small molecules as precision medicines. We've established methods to link genotype to phenotype, or in this case, biosynthetic gene cluster to genetically encoded small molecule. And the way we do this is this really elegant approach where we we identify self-resistance genes within these fungal biosynthetic gene clusters that enable us to predict what the target of that molecule will be before we ever isolate it. So this is all done in silico. Just to provide a little more context here on this resistance gene phenomenon, let's say we have some gem-producing fungus. It harbors some biosynthetic gene cluster in its genome that gives rise to some gem. Let's just call this some antifungal. So this producer can go ahead and ship this cytotoxic molecule out of the cell to kill off competing fungi in the vicinity and gain a competitive advantage. Because this gem has evolved to inhibit some essential target protein in these competing fungi. Now, in many cases, this producer and this competitor are going to be very closely related to each other. So the producer will often contain this very same ortholog of this essential target protein. So of course it needs some mechanism to protect itself from the cytotoxic action of this potent antifungal that it's making. One solution to this is the presence of these self-resistance genes that are often found within biosynthetic gene clusters that give rise to a given molecule. And we call these E-tags or embedded target genes. And these encode resistant mutant versions of these essential target proteins that enable this producing fungus to make as much of this molecule as it wants and kill off competitors without killing itself. So the reason we call this avatar RX is because these embedded target genes give away the target at the fungal level. And due to the high conservation between humans and fungi, these resistance genes effectively act as an avatar of the human target. So again, we can uh, we can carry out this target-centric approach to mine the fungal biosphere for gems that have evolved over millions of years to inhibit a specific therapeutic target. Just to summarize this first part of the talk, we've developed a scalable approach to mine these native fungal strains for the presence of self-resistance genes that serve as an avatar of the human target. We can then leverage genomics, transcriptomics, and metabolomics to identify suitable native production strains along with target-centric assays to confirm the desired biological activity, and ultimately isolate gems as advanced drug leads, which we can then iteratively optimize through medicinal chemistry campaigns to arrive at clinical candidates. Of course, isolating the gem, which we think of as an advanced lead, is really just half the battle. We still need to thoroughly characterize the biological activity of that gem in the desired disease context. For this, we can think back to the four pillars of drug discovery, where with any clinical agent or tool compound, we want to achieve exposure at the target site. We want to confirm binding to the intended therapeutic target, and we want to make sure that binding to that target is modulating the desired pharmacological and phenotypic response in a suitable disease context. And one could argue that target engagement is, in a way, the central pillar in all of this, because it's really the essential link between confirming exposure at the target site with the desired biological outcome in the disease context. And maybe I'm preaching to the choir here, but you could also argue that CETSA provides the most universal approach for addressing this issue of target engagement. We can do this in a quantitative manner to not only confirm binding against our intended target, but we can also carry out selectivity profiling exercises using mass spectrometry technology. And we can do this right in complex biological systems, as Renato alluded to. So we can do this in lysate, intact cells, even in tissue homogenate, for example. It's a really powerful approach as well because it's completely label free. So in contrast to certain other chemical proteomics approaches, we don't have to modify our molecule. We don't have to tag our proteins. This is all just the natural molecule right out of the fungus or some analog and some native biological context. So Renato already did a really nice job explaining CETSA, so I'll just go through this real quick. It really relies on this fundamental biophysical of proteins, whereas the protein's heated, it unfolds, denatures, becomes insoluble, and produces this type of melting or denaturation curve. In the presence of some drug or some ligand that binds to that protein, we can see either a stabilization or a destabilization event, which we can interpret as a proxy for direct target engagement. So if we think about this in a cellular context, with most drugs, the bulk of the proteome is not going to be impacted by that treatment. So there's going to be no stability shift. There's going to be a subset of proteins where their stability is either increased or decreased due to direct binding to that molecule of interest. And then since we're in living cells here, these things can metabolize. So there can also be certain other downstream biological changes that can impact the abundance readouts in the data. So this gives you a really comprehensive picture of what a given molecule is doing in a cell system of interest. The protocol here is outlined below. We can take intact cells or lysate, do a treatment plus minus compound, subject the samples to a heat shock, give them a nice hard centrifugation to pellet those insoluble proteins. We can then take that soluble fraction and subject it to a standard bottom-up LC-MS-based proteomics prep and analysis to obtain what's called this compound fingerprint, where we can identify destabilized protein hits and stabilized protein hits, which again, in a cell context, could be due to either direct target engagement events or downstream pathway modulation as well. So with that, let's dive into some of the details of this collaborative effort to study this polyketide lactone called Brefeldin A. This molecule was first isolated from the filamentous fungus Penicillium decumbens back in 1958, and it elicits very striking and profound biological activity, even within minutes of adding it to a cell culture. So it completely shuts down all protein export through the secretory pathway. And it does this by inhibiting this early anterograde step in protein trafficking from the endoplasmic reticulum through the Golgi apparatus. Not only that, within minutes, Brefeldin A causes the Golgi to start fusing with the endoplasmic reticulum, forming this sort of amorphous blob of an organelle. That's sort of reflected here in these confocal micrographs, where you can see with BFA treatment, this Golgi resident protein is dramatically dispersed through the cytoplasm. So really, really profound activity with this compound. And it's no surprise when you're looking at micrographs like this and completely shutting down the secretory pathway that this molecule does indeed elicit potent antifungal, antiviral, and anti-cancer activity as well. And Brefeldin A is a really nice kind of test case scenario for this LifeMind-Pelago collaboration because it's been very well studied and the mechanistic target has been well established to be this small GTPase called ARF1, which under normal conditions, this molecule in its GDP-bound inactive state can be activated by proteins called guanine nucleotide exchange factors, or GEFs, such as this GBF1 protein shown here, which catalyze the exchange of the bound GDP for GTP. This induces a change in the protein's conformation and potentially localization, and enables it to elicit these effects on Golgi morphology and protein secretion. And Brefeldin A does this in a really miraculous way. So it binds to this complex of GDP-bound ARF1 along with this guanine nucleotide exchange factor GBF1, and it locks these two proteins into an inactive complex through an uncompetitive molecular glue mechanism and prevents this GTP-bound form of ARF1 from carrying out its roles in protein trafficking. Here's a closer look at this molecular glue complex where we have Brefeldin A sitting in this nice induced pocket between ARF1 and, in this case, a fungal GEF protein here. So with all of these dramatic biological consequences with this molecule, we wanted to be mindful of this when we design our CETSA experimental conditions. So we want to try to keep the treatment time really short because this thing, again, dramatically alters Golgi morphology within minutes of adding it to a culture. And it can do this even at submicromolar concentrations as well. And we wanted to get a really nice comprehensive picture of the biological function of this fungal natural product. So we wanted to perform both a live cell CETSA experiment along with an analogous lysate-based experiment. And these have certain pros and cons, which collectively they kind of add up to the complete over here. So with the intact cell treatment, we can pick up direct target engagement events, which many times will elicit some sort of downstream biological changes on pathways associated with that target. So this gives us a really nice comprehensive view of what's going on, but by definition, it can then be difficult to deconvolute what proteins are showing a shift due to direct binding versus what's showing a change in abundance due to some pathway modulation event. So to shed light on this, it's always very powerful to supplement these live cell CETSA treatments with a lysate-based treatment where now we've disrupted the cell and the organellar structure. So we really take this pathway modulation out of the picture now, and we simplify this down to a more reliable readout of direct target engagement events. So looking at these two sides of the coin together can really get a comprehensive understanding of different molecules of interest. So I'll walk you through now this experimental layout for our pooled or compressed CETSA Explore experiment. And again, I want to thank Tomas here for doing all the heavy lifting and prepping these samples. And as you're going to see, generating some really cool data in the process. So for this, we chose a THP1 cell line because they're known to express good levels of both ARF and this GBF1 molecular glue complex partners of Brefeldin A. We can take these cells and then subject them to an incubation with different concentrations of Brefeldin A spanning a range from 80 micromolar down to 640 nanomolar. We can perform that treatment for half an hour at 37 degrees. And then we also included a nice quality control with a well-characterized kinase inhibitor, staurosporin, along with, of course, a DMSO vehicle-treated sample as well. And all of this is performed in triplicate. We can then take each of these samples and pull them out along a temperature gradient. So every sample now becomes 12 aliquots for this heat challenge where we have this matrix of increasing temperature and then increasing dose of Brefeldin A along here, along with DMSO and the QC sample. The typical temperature range for these experiments is usually in the neighborhood of 44 to 66 C. After this heat challenge, the cells are lysed through 3 freeze-thaw cycles. They are then pooled back together. So this is why we call this pooled or compressed CETSA. So they're combined back together along this temperature gradient. And this just simplifies the analysis because instead of looking at thousands of different melting curves for all the proteins in the cell, we've effectively reduced this to a more simplified differential abundance area under the curve type measurement. So after sample pooling, we give these samples a nice hard spin to, again, pellet the insoluble protein. We can take that soluble fraction and then prep that through a standard bottom-up proteomics type workflow. This is all done with a TMT Pro 16-plex multiplex format, where we have, again, 3 replicates of DMSO, 3 replicates of each of the Brefeldin A concentrations, along with the staurosporin quality control sample. Here's a quick look at some of the QC parameters for these data that were collected by Tomas and Pelago. You can see really nice deep proteome coverage here, over 8,000 proteins, almost 80,000 peptides, really nice low coefficient of variation here, which indicates excellent quantitative precision coming out of the mass spec readout. This box plot here also shows each sample. and it shows a nice symmetrical global median across the whole data set, again indicating really nice, robust, high quality TMT data. With that, let's go ahead and dive into some of the data, looking first at this volcano plot for Brefeldin A at the lowest concentration tested, so this 640 nanomolar concentration. Right off the bat, we can see destabilization of this guanine nucleotide exchange factor GBF1, which again is a well-established target of Brefeldin A. We also picked up these other two guanine nucleotide exchange factors indicated here, ARFGEF2 and ARFGEF1. Importantly, all of these proteins that are shifted here are associated with the Golgi apparatus. So this is really robust confirmation of the known biological activity of this Brefeldin A molecule. So many of these other shifted proteins here likely represent phenotypic effects due to these profound influence of Brefeldin A on Golgi morphology. Now, if we look at all four of the doses for Brefeldin A, here's the low dose we were just looking at. We also have 3.2, 16, and 80 micromolar. And it's really nice to see this thermal destabilization of these guanine nucleotide exchange factors occurring not only at the low dose, but also across all of these higher doses as well. If we take a closer look at this 80 micromolar top concentration, over half of those proteins, so 29 out of the 50 proteins that are shifted, are again associated with the Golgi apparatus, which is a really nice confirmation of the known biological activity of Brefeldin A. One thing that's interesting to note here, you'll remember I mentioned Brefeldin A stabilizes this GEF GBF1 with the small GTPase ARF1, so it locks it into this inactive conformation. We see a really nice shift for this GEF protein partner, but we don't observe any shift for the ARF1 GTPase. So it's detected in the proteomics data set. There's just no modulation there that would indicate a target engagement event. This type of behavior we've seen with other molecular glues as well. One possible explanation is that these two protein partners may have quite different melting or denaturation temperatures. So our standard gradient in this experiment may not have been suitable to capture this denaturation event for both of these protein partners. We can now take a look at these dose responses. So we've got different proteins listed here, and then the extent of destabilization along the y-axis and the Brefeldin A concentrations along the x-axis. And these are just all 12 of the proteins that were shifted at the lowest dose. And we can see this really nice dose-dependent destabilization for GBF1, ARFGEF2, and ARFGEF1. And what really jumped out at us is many of these other shifted proteins, so these other Golgi-associated proteins, many of these look saturated already at the lowest dose. So this likely indicates, again, this really dramatic and potent phenotypic effect of Brefeldin A on disrupting the Golgi complex. If we put this into a string analysis, looking first at the protein hits that were significant across all tested conformations, we can see all of these proteins are associated with the Golgi complex. So a very high degree of functional and physical connectivity here. And I'm just highlighting these primary targets, and many of these other proteins, if not all of them, likely indicate downstream phenotypic effects due to this profound Golgi disruption phenotype. If we look at this panel, this is now a string analysis of all the protein hits at the top 80 micromolar concentration. And here we're seeing the enrichment of the expected gene ontology terms such as secretion, vesicle-mediated transport, and regulated exocytosis. Again, very much consistent with the well-studied mechanism of this Brefeldin A molecule. And all the proteins colored in yellow here, again, are associated with the Golgi apparatus. So the bulk of these proteins, over half of them are Golgi resident proteins. Tomas also followed this up with a lysate experiment in THP1 cell lysate. And here the effects are actually quite striking. And there's a few interesting learnings if we compare these data to the cell-based treatment data. So right off the bat, we do confirm again a nice thermal destabilization of GBF1, but notice we're at really high concentrations now. So going from 80 all the way up to 500 micromolar with Brefeldin A to detect this engagement and thermal destabilization event. The statistical stringency was also relaxed here, you'll notice. So we're seeing in general a much less robust effect than what we saw in the intact cell context. And I think this is a really important point that for certain compound protein binding events, you really need that live cell context with the cell and the organelles intact to pick up that robust thermal shift signature. The other key learning here that really jumped out to us is that Brefeldin A looks like a remarkably selective molecule. So again, in these lysate experiments, we're typically looking only at direct targets of the compound. And even at 500 micromolar, we're not seeing much in the way of off targets here. And in fact, we can likely dismiss this phosphodiesterase 6D. That tends to be a common shifter in CETSA experiments. We can also likely dismiss this PEC7 protein because if we look at the dose response, it's only shifted at one of the concentrations, so that's likely a fluke. So really, we're left with just the known canonical target, this GBF1 guanine nucleotide exchange factor, along with this thioredoxin interacting protein TXNIP. So this is why genetically encoded small molecules or natural products are so incredibly attractive to us at Lifemind because they do tend to demonstrate this really exquisite selectivity. And again, that's just coming from years and years and years of evolution to selectively and potently bind a given protein in the cell. So with that, I will go ahead and wrap up. I think this was a really nice collaborative effort between Lifemind and the folks at Pelago. We were able to nicely confirm the expected engagement of Brefeldin A targets along with this pronounced Golgi disruption phenotype. And as I mentioned, we see a much more striking effect in live cells compared to the lysate. And this, especially in the case of Brefeldin A, it's not hard to imagine a scenario where you really need the organelle structure for the ER and the Golgi to be intact, for some of these ARF and GEF proteins to be localized at the right part of the cell. So I think we've demonstrated that this CETSA Explore methodology is a really powerful approach to characterize not only these fungal gems, but of course any molecule of interest as well. And we're in the process of taking this collaboration with Pelago even further to profile the selectivity and confirm target engagement for molecules coming out of one of our internal programs as well. And I think that's already been hugely instrumental in helping to optimize that molecule as we try to convert it into a clinical candidate. And I just like to thank the folks at Pelago, namely Stina, for the invitation to come give this talk, along with Tomas for generating all of this really nice data with Brefeldin A. Alexi as well, he was very much involved in the initial planning of the experiment. And finally, thanks to Jacob and Bora again for organizing this event as well. On the Lifemine side, I'd like to give a big thanks to Paola Castaldi, who was my previous boss at Lifemine. She unfortunately left a little while ago, but she was hugely instrumental in, again, giving me the opportunity to make this talk and also in helping to design the experiment. I'd like to thank all the members of our chemical biology and proteomics group and the bioinformatics team for some helpful discussions. And also Greg Verdine, one of our founders at Lifemine, who really took a nice interest and gave some really nice input into the design and results of this study as well. And with that, I'm happy to take any questions.
Okay, thank you so much, Ben. This was a great presentation, a great illustration of how Pelago can actually help in the drug discovery process. I think we can open the Q&A session. So if you have any questions, you're welcome to just type them in the chat box, and I will ask them to Ben. I'll read them. In the meantime, and while people are just warming up to come up with a few questions. I had a question, like you mentioned, that you're basically using the fungi and they are more close to humans than actually plants are. Do you see more potential in finding new medicines for humans in the fungi that already have some sort of relation with humans, either parasitic or symbiotic, or maybe in the ones that don't have any relation, maybe mushrooms or
Yeah.
so, or is there any difference between these two? Do you have any comment on that?
That's a very good question. That's not something I've thought a whole lot about. I think you make a great point, though. There's a potential opportunity to even think about fungi in our resident microbiome and the potential to sort of mine some of those molecules that have potentially already evolved not to modulate some fungal process or some bacterial process, but actually our very own biology. I think that'd be a really cool thing to try to get into. Unfortunately, we haven't really launched any efforts along those lines yet. And on the other side of that, your question about mushrooms, we're also not looking at those at the moment. We're exclusively focused on filamentous fungi just because they have these large strain collections available, and we don't have to worry about conditions to try to induce, mushroom fruiting formation and stuff like that. So, but again, there's a huge opportunity there. And actually, I think lovastatin is produced by oyster mushrooms as well. So it's not only in the filamentous fungi, but even in higher fungi as well. And that's probably the case for many of these molecules.
Okay, very good. We already have a few questions on our chat box. We have from Christine Jones. Can you comment on the compound incubation time and compound concentration with the live cells? Did that require substantial optimization, or are there guidelines that you tend to start with?
Yeah, there's certainly guidelines that I think are going to work in most cases. So generally you're going to see anywhere from a half hour to an hour treatment in a live cell context. And it's always beneficial to shoot high on the concentration because you really do want to saturate the target in order to detect a nice robust thermal shift. So there's always those two considerations. It can be helpful to do a dose response as well because as you've seen, we can show a nice dose dependent shift, but it's definitely helpful to at least have a couple of the concentrations be on the higher side of what you might normally use for a certain molecule. In this case, with Brefeldin A, um, due to the profound effects within minutes, we were trying to keep that incubation as short as possible. And again, we still settled on just a 30-minute incubation without any other sort of optimization. And, it still, I think, worked very well in this case.
Okay. There are more questions coming up. So from Juan Gutierrez, any thoughts on the effects of BFA on TMED proteins? Are they direct targets or just pleiotropic markers?
TMED, yeah, good question. Yeah, I still think these are unlikely to be direct targets. Again, they are associated with the Golgi, so they're probably just being caught up in some of this collateral damage with the really dramatic morphological changes that happen when Brefeldin A engages these ARF proteins and these GEFs at the Golgi membrane. And
Okay.
again, I guess the other thing that speaks to that is we don't see any of those proteins showing up in the
Lysate.
lysate experiment.
Mhm. Great. Then Alexei is asking, 2 out of 3 fungi-derived compounds are molecular glues. Is it a common trend in GEM's mechanism of action or just a coincidence?
I think this is something I've wondered about a lot is how does this break down? I know our our CEO Greg Verdine, he likes to think, and he may very well be right that these molecules are biased toward molecular glue type mechanisms. I don't think we really know enough to say. These just happen to be the examples I chose for this presentation, where 2 of the 3 were molecular glues, but it's definitely something that I've thought about as well. Because I mean, molecular glues are very interesting in that they can be these crazy complex molecules like cyclosporine or tacrolimus or Brefeldin A, but then they can also be these relatively simple, imide compounds that were discovered kind of serendipitously.
Great. Then Beth Hoffman, do you have a sense of how potent the compound needs to be in order for CETSA to work?
Hmm. I think if you have something with micromolar type affinity, you have a pretty good chance you're going to see a shift if the target's expressed. That would be my guess. We don't have any real empirical data. Pelago or Renato can probably maybe speak to that a little bit better than I
Yeah,
can.
if I can step in on that. So usually we have as a rule of thumb that we like to saturate the system, and that means we should go a bit higher than IC50s you get from other cell-based assays. Going higher means going maybe 1 or 2 logs above, which means that something within the micromolar range will still be easily captured. And as you can see on the slide that Ben is sharing right now, uh, we went to a very, very high concentration, 500 micromolar. It's really a very high concentration. We don't usually go as high for most compounds. So it's a matter of trying. If you have more information, if you would like more detailed analysis, you're welcome to contact us, Pelago. Either drop us an email directly through our website or book a meeting directly there, and then either me or one of my colleagues will certainly help you with analyzing your specific case. Um, Another question that we have here from Vic C is: are the live cell versus lysate observations with BFA generalizable to other molecules, or do you think this is very specific to BFA?
I guess I can answer that kind of in two parts. I think it is very characteristic where a molecule may show many more shifts in the intact cell context due to these downstream biological modulation events in addition to the target engagement events. Whereas in the lysate, we kind of take this downstream modulation out of the picture. I would say, and again, Renato, you can correct me if I'm wrong, but I think it's this situation with Brefeldin A where we have to go to such high concentrations in the lysate to capture this shift. I think that is relatively rare. At least in my much more limited experience, the doses tend to be similar to capture the same primary target engagement event in both a lysate and a cell context. And again, I think part of the intricacy with this Brefeldin A experiment is that it is disrupting the Golgi in such a profound manner. And some of these ARF proteins, they need to be, for example, lipidated and so forth to localize to the right part of, the Golgi complex. I think that's playing a role here.
Actually, Alexei also stepped in on the chat, and he mentioned that we have observed similar patterns with emids, and he
Mm,
posted a link to one of our
cool,
publications. So uh, everyone is also
cool.
welcome to look in the chat and read that publication. We don't have any more questions, but we are welcoming any questions that anyone from the audience may have. But in the meantime, maybe I can ask if you didn't know that GBF1 was already a target before you started, would you have changed anything on the layout of how you run the experiment?
Um, probably not, other than making sure we're picking a suitable cell line where we've observed some sort of biological activity with the compound to make sure that we're in the right sort of biological context. And again, many cases, you obviously won't know the target. So in any CETSA experiment, you really have to go back through the mass spec data and do a targeted approach on a Western blot or so forth to really confirm if those are truly targets and that shift is real and follow up with a dose response and all that good stuff.
Yeah, and that, if I may add, is is the beauty of this kind of assay, which is completely unbiased.
Yeah,
So the fact that we know some of the targets will not change, in principle, the way that
yeah.
the assay is conducted, because we want to have a completely unbiased analysis and see the whole proteome effects without biasing the system in any way. Um, I don't see any more questions coming right now, but maybe. Um, the GBF1 appears as a destabilization, but the mode of action that you showed suggested that it should be stabilized in a complex with BFA. Um, do you have any explanation for this?
Yeah, that's something that we've grappled with as well. That's a really great observation there. It yeah, it's so I think one way to potentially explain this is maybe this kind of unnatural complex that Brefeldin A induces between ARF1 and GBF1 that may, in some sense, destabilize GBF1 relative to when it's either alone or in kind of the native natural context when it's binding to ARF1 to actually catalyze this nucleotide exchange event. So the complex is Brefeldin A sort of intercepts this complex of GBF1 and ARF1 when GBF1 has essentially bound to ARF1 to perform this nucleotide exchange event. It intercepts that compound and changes the conformation slightly to inactivate it so that it, yeah, we don't know for sure, but that would be, I think, one possible explanation is that the stability in that this BFA induced complex is different.
Yeah, if I could add a comment on that, I would say that the stability that we are looking at on this slide is a biological stability. So you're basically stabilizing the protein in the biological context. You're not you're making that the protein is no longer active or somehow arrested in a certain state. Whereas the stability we talked about when we are doing a CETSA analysis is a thermal stability. So it's something that happens with the increase in temperature. It's a biophysical property of the protein and should not be confused with the stabilization that occurs on a biological level.
Sure, yeah.
Okay, I don't see any more questions coming up. I think your presentation was spot on, clear. Everything was very well explained. Congratulations
Thanks.
on that. It was a
Appreciate
pleasure
that.
to be able to host this. If there are no more questions coming up on the chat, I think we could wrap this up. Just let a final word thanking to Ben for being available to actually conduct this webinar for us. Also, everyone at Pelago that has been involved in the organization, and always good to leave a note for everyone interested in knowing a little bit more about CETSA or maybe understand how CETSA can be used in your own projects. You're welcome to go to our website, pelagobio.com, where you can book a meeting directly with one of us to actually get a personalized consultation or a personalized discussion on the specifics of your project and see how we can help you either with CETSA or any of the other complementary assays that we can add on to that. So if there is no more questions, thank you very much, Ben. And I hope to see you all on the next webinar coming soon.
Yeah, thanks, guys. Thanks for listening.